I'd like to acknowledge that we're meeting on the lands of the Bedegal people, who are the traditional owners of the lands that this campus has been built on. I pay my respects to elders past, present and emerging. I'm Christina Feinler-Bissett from the University of Adelaide, and for those of you who don't know me, I'm the current president of ANGMA. It is my great pleasure to welcome you to our 2023 ANGMA meeting, the second one really in a year, which is an amazing feat, I think, for our small society. We've got an amazing program to look forward in the coming 1.5 days after already experiencing what was an outstanding public lecture event last night. And I would like to take this opportunity again to thank the 3 speakers, Professor Imad El Omar, Sunny Wong and Nick Talley, for their outstanding talks and their generosity in answering many very interesting questions from the public at the end. And these questions from the public really indicate a strong interest in our field, and therefore, to me, just reinforces the importance of engaging with the public as a professional society as well. So this was an excellent initiative. A meeting like this would not be possible without the hard work of a very dedicated organising committee, as well as generous sponsors. And so I'd like to wholeheartedly thank the local organising committee under the very capable guidance of Michael Sesniak and Peter, over there. And they were supported by Alvin Chia, Nick Talley, Santosh Sanagapalli and Mei Wong, and really worked tirelessly to put this meeting together. And according to Peter, I think Michael practically had a full-time job on his hands in the last month. And as a result, we have a very exciting program to look forward to, and in what a beautiful location as well. So thank you very much to you all for your dedication and huge effort. I think that deserves a round of applause for the organising committee. Our sponsors include Dr. Falk Pharma, Biome Bank and Medtronic as platinum sponsors, and Takeda and Bayer as gold sponsors. Thank you all for your generous financial support. It's no exaggeration when I say that it would just not be, we would not be able as a society to offer this meeting without your support, so thank you very much. Finally, I'd like to thank all of you in the audience for attending the meeting. While the meeting is for you, it's really also happening because of you and your varied contributions, be it a poster, an oral presentation, an invited talk, volunteering in the live demos this afternoon, or asking questions at the end of talks in a session. All these contributions help to generate the best possible meeting for all of us. So once again, and also on behalf of the entire ANGMA committee, thank you all for being here, for your support and for your hard work. Welcome to ANGMA 2023. And I'm just handing over to Peter just for a couple of housekeeping.
All right, thank you. Just very quick housekeeping issues. Firstly, there is a university exam upstairs. So, I'm sorry. So please don't hang out and speak too loudly in the foyer. Please, during the break and lunchtime, please try to stay inside the gallery as much as possible. There should be plenty of space in there or outside of the building. And at the end of each session, one of us will be traveling up and down with microphones for you to ask questions. Okay, thanks.
Thank you much, Peter and Christine. And good morning, ladies and gentlemen. We're absolutely delighted to see so many of you turn up this morning. I'd like to thank Michael and Peter for putting on an excellent program, which I hope will be stimulating and a further inspiration for more work in this very fast advancing field. So we have a wonderful program. This session will have 3 outstanding speakers. I have great pleasure introducing the first one. This is Professor Georgina Hold, my dear friend from 22 years. We started together in Aberdeen, 2001, and stayed together. And I'm absolutely delighted to welcome her here. She's Professor of Gut Health at the University of New South Wales. She's also the Director of Research for South Eastern Sydney Local Health District, and one of the giants in the microbiome field, and particularly in relation to inflammatory bowel disease, although today she'll be talking about bowel health from within. The title of her talk is Harnessing the Power of the Microbiome. I think she'll be talking more about therapeutics. Thank you, Georgina.
Sorry, I've got someone else's slides. I can do your talk if you want, Eunice, but... That's not mine. Oh. Okay. So, good morning, everybody, and thank you so much to the organisers for giving me the opportunity to come and talk to you today. Thank you so much to Peter and Michael and to ANGMA for having the meeting here. I'm sorry that I couldn't be with you last night to hear the public lectures, but the microbiome was calling, and I was in the lab trying to finish some stuff. So, and I gather that I missed the weather. So. I hope everybody enjoyed the Sydney weather. So, as Prof. Elamar said, my interest is inflammatory bowel disease, but I'm a clinician. I'm not a clinician, and I come to the microbiome as a basic scientist who's always been fascinated about how bugs cause disease, and through that interest, it really has shown me that they may cause some of the diseases and some of the pathological processes that we try to solve, but actually they give us the clues and probably the solutions to how to fix these conditions as well. So Prof. Elamar and I, as he said, has been a partnership for the last 21 years. Before I had the opportunity to meet Prof. Elamar, I was undertaking my PhD where I was essentially defined as a microbial ecologist. So that was just 25 years ago where when I got my PhD last century. Microbial ecology was what you studied if you wanted to look at microbial communities and relationships between them. 25 years on, I haven't changed, my interest hasn't changed, but I'm now a microbiome expert. So it just really shows you how things have changed, even in the last 2 decades. And it's really interesting to think. The last few years has shown us how quickly we can do things now where we couldn't do them before. So the development of a COVID vaccine in 10 months from bench to bedside, for example. But really, when you're talking about the microbiome, and this is why Prof. Elamar set up the Microbiome Research Centre here within the University of New South Wales at St George Hospital, is because there's so much, and it levels everything really. Everything kind of comes back, as he would say, to the gut. I would say everything, and I suppose we would both now say everything comes back to the gut microbes, but it really does shape what we're doing. So apologies if any of this was gone over yesterday in the public lecture. The microbiome, what we have associated with us, if you just looked at it from a cellular perspective, 90% of your body is actually comprised of cells that are not yours. So think about how much that can influence all the various processes that we're doing. If we look at this at the genetic level, at the genome level, actually that increases to 99% of the genomic content associated with your body is not actually yours. So the microbes are this ecological community of commensal, symbiotic and pathogenic microorganisms that literally share our body space. The one thing to take away from this is they are not innocent bystanders. And so this concept of the microbiome probably most effectively aligned to the paper that Joshua Lederberg wrote in 2001, but again shows you this is where we've gone in less than 3 decades. So the gut plays a central role in health, but the gut is a sum of parts. It's that alignment between the host immune system, the microbes that are in the gut, and putting that kind of neurogastro angle on this. 70 to 80% of immune cells associated with the body are in the gut. Over 100 million neurons are in the gut, and these are producing factors like neurotransmitters. And you know that the majority of the body's serotonin is located in the gut as well. So to think that there isn't a link between the gut and your brain would be churlish. What we used to consider in terms of the gut being essential for digestion and absorption, metabolism, has more recently moved to understanding the impact of the gut in terms of satiety, functional consequences, and then very much more recently, as we've understood more about the gut-brain axis, it's been moving on to look at cognition and psychological effects. So the gut-brain axis is not a new concept. It's this bidirectional signaling between the gut and the brain, but also the brain and the gut. It's been demonstrated in the context of the gut microbiota, which is the newer concept, the gut microbiome brain axis, that without a microbiome, your homeostasis is dramatically changed. And this has come from the germ-free animal studies where we see blunted responses in terms of cognition. We see delayed gastrointestinal transit times, and we also see ENS abnormalities. And so this really came to, and if you haven't, if this interests you, if this area interests you and you haven't looked at anything, I would say go and look at this gentleman, John Crean from APC in Ireland, who really is the forefather in understanding the gut microbiome brain axis. And John writes beautifully, and this is from one of his papers back from 2019. It's a tome, but it's a really, really enjoyable tome to read, talking about all the different factors in relation to the microbiota gut-brain axis. But what this shows you is that the microbiome is impacting a number of neurodegenerative disorders, metabolic disorders, and also functional disorders and psychiatric disorders, also playing a very strong role in pain and in anxiety and stress, and also in the functional gastro disorders such as IBS. But one thing that it does show us is that not all microbes are equal. As I was saying to you, it really does demonstrate that they are showing they are part of the problem, but they're also, and this is what I'm going to talk to you about today, hopefully part of the solution as well. And so this concept of targeting the microbiome, using novel therapeutics that are going to target the microbiome is really what has come out of the work in the last 2 decades. And this concept of psychobiotics, which is essentially manipulating the microbiome using factors that are going to improve mental health, is really probably where my interest and the interest of this meeting actually align. So what we know about microbes in the gut, we know that stress and some of the neurotransmitters that are produced in the gut impact on microbes just as much as microbes can produce some of these neurotransmitters such as gamma-aminobutyric acid, but also somatostatin. And we know that all of, just because some microbes are there, different species of those microbes don't all do the same thing. So just because you understand what one E. coli does, for example, E. coli Nisle, which is one of the probiotics that we use that shows some improvement in IBS, produces some of these neurotransmitters, but other E. coli, such as 0157, which is a pathogen, it doesn't have the capacity to produce some of these things. So we really have to bring the microbiology or the microbiome science along with us when we're trying to understand what it is that we're trying to achieve. We also know that various microbes produce virulence factors, such as toxins and autoinducers, and actually the numbers of microbes that are present in the gut can also shape what's going on. So it's not just about knowing who is there in your gut, it's about understanding what they are capable of. And this really brings me to the three factors that I want to talk to you about the microbiome in three. We know now that it's functionally important. Functionality of the microbiome is much more important than who is there. We know that it's unique and it is affected by your environment, it's affected by your genes, it's affected by your diet, it's affected by everything that we do. The fact that we all came to UNSW main campus today and had coffee from a different cafe, is going to have some kind of impact on what's going on. But ultimately, it's modifiable. And it's also modifiable by things that we, as the public, as well as the medical profession and the scientists, can do. So we are able to have an influence on our microbiome by just understanding more about what causes the microbiome, what makes it tick. So what we eat is what they receive. So I'm not here to advocate that we should all not eat the things that we like, but there really is a place in the future for us to be able to proactively influence our microbiome for the better. So microbial therapeutics, and what we're talking about are factors that are altering the gut microbiota to improve health. And we know that this is well established, and we've been doing it for a long, long time. We just didn't know that that's what we were doing. The microbiota, thousands of different species there. For the majority of what I'm going to talk to you about today, we're talking about bacteria, but that's a very, very simple overview of the microbiota. And this is another of those technical factors. We're very good at dealing with bacteria. The others are a wee bit hard, and we haven't quite got the technology or the infrastructure to do it, but my golly gosh, we need to understand these things as well. But for today, the majority of what I'll talk about is bacteria. So we have the conventional concepts in terms of microbial manipulation. And when these were set up, they weren't really set up with the idea that they were changing the microbiome. They were just actually improving health or making us feel better. So we have what we class as a kind of dietary interventions. We have the prebiotics, which prebiotics are foodstuffs, stuffs that bacteria like to eat. So it's the things that bacteria can digest lots of different foods, and they'll do different functions based on what it is that you give them. So if I gave you a McDonald's or I gave you a celery stick, you're going to eat them both, probably. One you'd prefer to eat over the other, but you're going to do something very different with that nutrition that you've taken. Bacteria are just the same. Give them what they want. They'll give a whole repertoire of genetic factors, functional factors. If you only give them one, then they're going to kind of shut down various processes because they're not very happy. So give them the things that do the beneficial things, and you'll then get a better outcome. It's like feeding your kids, isn't it? Sugar. Do you want them to shut up or not? Probiotics. Probiotics is the actual microorganisms. So this is your Yakult, your Danone, your fermented milk products that we're actually giving live organisms. If you want to give them both together, which kind of makes sense, if you put the stuff in that you want with the bugs that you want, then we call that a symbiotic. And then also they lump antibiotics into dietary interventions. We're not going to talk about antibiotics today. The other sort of more novel concept, the emerging concepts in terms of microbial manipulation, your fecal microbiota transplantation, phage therapy, live biotherapeutics, and also microbiome mimetics. So these are all the things that are coming. They're in different shape of readiness for the various different clinical disorders. And I have to say, in terms of neurogastro, very much on the infancy side of understanding the importance of these. So I've talked to you a little bit about probiotics and prebiotics. Probiotics, and the definition for them is live microorganisms which, administered in adequate amounts, confers a health benefit on the host. Usually, these will be in the guise of bifidobacteria or lactobacillus species, your lactic acid-producing bacteria. Why? Because we could grow them, quite honestly, because they did do something good, but actually we now know that they're not the ones that we need to be focusing on. They don't, a lot of the time, probiotics are supposed to colonize, to then confer their benefits. Quite often, particularly, the data from IBS studies would say as soon as you remove those probiotics, the symptom relief goes away. They are there to reduce pathogenic load, and they do that through competitive exclusion, and we do know that they're capable of improving the mucosal barrier integrity. So they do lots of good things, but take it away and it's gone. So that's where the argument for something like a prebiotic would come in, or a combination, because that can then keep the ones that are there happier. What we know in relation to neurogastro studies is that the use of certain bifidobacteria and lactobacillus are known to decrease the pro-inflammatory mediators, such as your pro-inflammatory cytokines, reduction in your NF-kappa B signaling, for example, increase anti-inflammatory effects, such as the increase in polyunsaturated fatty acids. They're also known to be able to impact on GABA circuitry. A number of bifidobacteria and lactobacillus can produce GABA. And they have also been shown that ingestion of probiotics can regulate stress-induced behaviours. There are a new age of probiotics coming. They already exist and are used in conditions such as obesity and metabolic disorders. To my knowledge, they are not present within the kind of neurogastro environment at the moment, but we are still at the very early stages of understanding these, and these are those microbes that we've only really been able to culture in the last 10, 15, 20 years. Because the gut is an anaerobic environment, so any oxygen coming into the system means that those bugs die straight away. We know that some of the most important of those bugs, the ones that produce the good things like short-chain fatty acids, which also impact on the circulatory systems, 30 seconds exposure to oxygen and those bugs are dead. So we've really got to make sure that our processing capability to develop these new age probiotics actually keeps track with what's actually needed. So fecal microbiota transplantation, I'm sure I don't need to explain the concept of that to anybody in the room, but there's a lot of information out there about the use of fecal microbiota transplantation. And again, this whole concept, which came from the recurrent C. diff story, in which FMT was actually improves, is a 98% success story, to the point that clinical trials were stopped when it went head-to-head with antibiotics. So we know that it's great for C. diff. But in C. diff, FMT, which is introducing donor stool into the intestine, however it's done, whether it's orally or through capsules or through an endoscope, it's targeting a monoinfection. It's quite an easy thing to do from a microbiological perspective. Get that one thing out with and replace it with anything, and it will improve. I know that from a clinical perspective, that's a difficult concept. But, FMT has been absolutely transformational in C. diff, but using that concept, that blunderbuss approach to putting an FMT to solve all clinical conditions has really not worked out very well. So studies in IBD, for example, are 40% success, no different to our standard treatments, pharmacological treatments at the moment. Why? Because the disease is a very different type of disease. It's a field defect. You can't just look to boot one thing out because there isn't a single microbial cause of inflammatory bowel disease. Going back to that whole, how do we make FMT? We stick stool in a blender and then add it and give it to a patient. We have to think more cleverly. We have to think more targeted for using things like fecal microbiota transplantation. And that's going to be no different than in neurogastro. as well. The studies in IBS, for example, only show a 22% better response rate of using FMT versus placebo in IBS, showing that probably we haven't thought enough about the clinical cohorts and we haven't thought enough about how that FMT needs to be prepared or delivered. There's also the concept of autologous FMT coming in, which is to give a patient back their own donor stool. So that concept of could we bank stool when somebody is well, to actually give them back their own to improve engraftment. And that's something which is being talked about. That's certainly something that John Crean is talking about in his papers. So just to wrap up, things to consider in microbial therapeutics. What is it that we're actually trying to do? We need to stratify cohorts more, because if we don't, then all we're doing is applying a microbial therapeutic by blindfolding ourselves and actually trying to hit a moving target. What are the organisms that we need to put in there? What are the functions of the organisms that we're actually wanting to enhance? Do we need to help the fecal microbiota transplantation or the selected strains? Do we need to pre-treat? Does antibiotics play a role in helping us to actually be able to hit that target? How are we going to give these selective therapeutics, and how are we going to measure success? All these factors are going to help us to move forward to improve health. Bugs and drugs, very last thing, 2 slides on this. Knowing that we can manipulate microbes to improve health, we also need to remember that bugs impact on the drugs that we use, and the drugs we use impact on the bugs that are there. So this is something which we've understood. We've been using this information for a long time. For example, in the use of sulfasalazine in IBD, we use the microbes in the gut to cleave the prodrug into the active form so that we can actually reduce inflammation. However, there's been a lot more work in the last 5 to 10 years trying to understand what all drugs do against all bugs. And this is a classic example here showing you where the bugs that bioaccumulate, they don't even biotransform, they just accumulate the drugs, so they take them out of the circulation so that they're not available to us as humans. But those bioaccumulators then produce their factors that then impact on other microbes that are also there. So depending on who's there, some are going to suck away the drug, and some of them are going to react to the fact that the other one sucked away the drug and do something different. So this is just showing you, in the context of one of those drugs, duloxetine, which is used in anxiety and depression, what happens with or without the presence of the drug in a microbial consortium of 5 common microbes, where we have Streptococcus salivarius, which accumulates duloxetine, which then changes its profile. It puts out microbial products which actually stop another of the microbes, Eubacterium rectali, from actually growing. And so this just demonstrates to you, in the presence of duloxetine, Eubacterium rectali, the red one, stays in high numbers. But if the duloxetine isn't there, Streptococcus salivarius doesn't do its thing, and then Eubacterium rectali can't respond or doesn't have the factors it needs to move on. So we really have to understand the communities of bugs if we're going to get to an understanding of how we can manipulate them for health. So current therapeutic options are still crude, but in many cases pretty effective. The second of the slides is where we actually need to go. It holds huge promise. We need to personalise. We need to understand the conditions. We need to stratify cohorts. We need to understand from a personal perspective, fine-tuning so that we can target and deliver personalised medicine to improve health. And I'll leave it at that, and I'm happy to take any questions. Okay, questions. Shameless plug. Anybody interested in doing research, wanting to be a healthy control, scan the QR code and join up for a microbial monitoring study which is going on.
Thank you very much, Professor Virginia Ho. I would like to introduce our second speaker of this section. So Dr. Rohan Williams obtained his PhD in intestinal motility at the St. George's Clinical School in 2003 under the supervision of Professor Ian Coe. He then moved fields into the bioinformatics, genomics, and system biology as an NHMRC Peter Doherty Fellow at the UNSW, working with the geneticist Peter Little. And then from 2007 to 2011, was a group leader at the John Curtin School of Medical Research at the ANU. And then in 2011, he moved to the newly formed Singapore Center for Environmental Life Science Engineering at the NUS Singapore to develop the metagenomes and system biology approaches for microbiome and microbial ecology. So he is now a principal investigator at the Singapore MIT Alliance for Research Technology. It's got a very good name called SMART in abbreviation, and also a scientific co-founder of the Blue Maiden Biosciences, a microbiome-anchored Singaporean biotech company. So his talk is about the metagenomics and multiomics for analyzing microbiome. So 20 minutes to the horizon. So please join me to welcome Dr. Johan Williams.
Thank you very much, Howard, for that kind introduction. In my talk today, I just want to sort of survey some of the major approaches that are being used to study microbiomes from both a compositional and functional point of view. And a bit of a disclosure, I'm the co-founder and equity holder at Blue Maiden Biosciences. We are a company that our long-term goal is to develop new therapeutics, develop new small molecule therapeutics from human microbiomes. Very happy to have Nick Talley as one of our SAB members, among other authorities, and Damien Keogh, the founder and CEO, is here at the meeting today, so very happy to discuss our activities with anyone. If we need to be reminded, and I think Georgina made great reference to the concept of microbial ecology. Microbiomes and complex microbial communities, of course, are important outside the human context and certainly beyond the gut-stool microbiome. So this, of course, includes in animals and plants, other micro-eukaryotes, and also in environmental settings and in engineered environments like wastewater treatment plants and various types of food. And a lot of the techniques I will discuss have been developed in other contexts, non-human contexts, but are also applicable to them. It's a very powerful set of shared methodologies. And really the major problems that are faced, I think, in this area are actually quite simple. At the end of the day, we really don't know a lot about most microbial life. We don't know how to culture most of these species. The fact that we can't culture them means that we can't employ the classical repertoire of genetic or biochemical, physiological, morphological techniques to study them. We don't know what many of these species are doing at a physiological level, and we're just beginning to understand, I think, and that's come out beautifully in the presentations in this meeting, is how they contribute to health and disease states. And so developing new ways of culturing microbes is really still very important, irrespective of anything I'm going to say today, and I think culturing is undergoing a kind of fascinating renaissance. The things I am going to talk about are basically culture-free microbiology based on DNA sequencing and other types of molecular phenotypes, how we recover genomes of these member species of microbiomes, and some new ways to study their function in situ, which I think the last speaker kind of really beautifully brought out at the end of the talk. And so we're just going to sort of pose this as a series of questions. The most basic question is, if you have a microbiome, what species are in it? And what could they potentially do? What biological functions can they perform? And we address that question using shotgun metagenomics. You could also use 16S sequencing. If we want to consider what functions the microbiome is actually expressing or contributing, we need to use a different set of techniques, whole community RNA sequencing, or maybe whole community proteomics. And then if we want to consider the broader contribution to the physiology of the organism, we could sort of start thinking about using metabolite profiling. And of course, I'm not mentioning the use of any other data here. Of course, that's absolutely critical. Clinical phenotypes, imaging data, or any other type of data that might be part of a broader study. This is purely focused on omics. And 16S amplicon sequencing has been used for many years. This is a landmark paper from the 90s in the context of microbial ecology. It is still a very, very useful technique, but if you're looking at getting into this area, please consider that it's really only, you can capture taxonomic information about what species are there, but you don't know anything else about the functional capacity of the organism you've discovered. You don't know anything about the other genes that are there and what those genes are doing. And so I think the approach that has emerged to sort of is going to become dominant in this field is whole community shotgun metagenomics. And so what's happening here is it basically taking a microbiome sample, extracting all the DNA from it that we can, and then that DNA is sequenced. If we were going to do ribosomal amplicon sequencing, that would be a separate workflow from the position here. So the DNA is sequenced en masse, and then if you come down to this part, is the cursor visible here? Yeah, probably not. Yeah, we can just see the light. So we sequence the DNA. These are massive data sets, and then what we want to do from this is obtain various types of information. We certainly want to understand who is there, what species are there, and what they're doing, and then also capture some very specific information about the nature of their genomes and the genes that are encoded on those genomes. And there's several major ways that this is done, which I'm going to just unpack now. And so the first way is what we call read level analysis. So in this case, we've extracted DNA, we've sequenced it, we've got these days this will be like per sample. This will be tens of millions or hundreds of millions of these short DNA reads per sample. They're 100 or 150, 300 base pairs long. And the classic strategy, and this is implemented in many bits of software now, is to take each of these and you map them against a reference database, and then you try and figure out what species they're coming from, or what genes they're coming from within that genome. And the advantages of this is that it's actually it's readily interpretable, and it's very, very open to quantitative analysis. So you can tabularize this data very easily. You've got essentially you're looking at the same thing across many, many samples, and it's very easy to incorporate this into study and broader study analysis. But the disadvantages are that ultimately, if you use this approach, you're dependent on the reference genomes in a database being the same as those in the community under study. And that may or may not be true. For the human gut microbiome, I think we're approaching the position where that is a pretty realistic goal. And a corollary of that is that you're only detecting known genomes. If you're dealing with a novel genome, you won't detect it. There's a high false positive rate in taxonomic assignment, and it's difficult to infer what I'd call the ecogenomic context. So it's not really obvious with these techniques whether you have one instance of a species or sort of 4 or 5 subspecies, for example, in that community. And there's a really good case study, actually, I think, of the limitations of read level analysis. And in this study from a few years back, this was a massive metagenome study of surfaces on the New York subway. And so there were several thousand samples collected. They were shotgun sequenced, analysed using read level analysis. And one of the very controversial conclusions from this study was that bacteria that caused bubonic plague and anthrax were commensal members of the New York metagenome subway. And this got hit very, very hard by a group of epidemiologists and genome biologists and so on. And basically, the argument was made that while these species were detected in this kind of taxonomic profiling exercise, there was no evidence that and it's quite a nice phrase, that the backbone genome of these organisms was actually present in this analysis. And so I think this is it was a very important study and a very important correction that a lot of us working in this space took notice of. And so so an approach that's using the same data but tries to solve this sort of limitation is what we call metagenome assembled genomes, or MAGs. And so what we're doing here is using exactly the same shotgun sequencing data, but instead of treating these reads as the unit of analysis, these are getting assembled by a computer algorithm into longer stretches of interconnected reads that are called contigs. And then you can do a few more tricks, and you can put these together into longer pieces again that are called scaffolds. And contigs and scaffolds are basically fragments of genomes of the species that are in the community that you're measuring. And the details of this get a bit turgid. There's a kind of summary here on the left. But what we're trying to do is capture whole genomes, and ideally that would be a continuous DNA sequence that would be 4.5, 5 megabases in length. But in reality, what we're detecting is something more like this, where the genomes are highly fractionated. There's like tens or hundreds of contigs, and we have this problem of figuring out which bits are missing and which bits are contaminated from other non-cognate genomes. And there's a framework that's emerged for this over the last sort of 5 to 10 years. These analyses are very, very powerful. For example, this is 913 metagenome assembled genomes from a cow rumen microbiome. This is work from Mick Watson at the Roslin Institute. And it still kind of blows me away with this stuff, that you get these phylogenetic trees, you can do taxonomy on these whole genome sequences now, you can figure out what species they actually are. Each data point on here is actually a whole genome or some approximation thereof. But one thing that I think is underestimated in many reports of this kind of analysis is that actually most of the genomes you will get out of this exercise are actually of unremarkable quality, particularly in complex communities. Like so for the human gut, for example, or something more complicated than that, a lot of environmental samples. And you can see here as the abundance of a species decreases, the proportion of genomes we're getting that are high quality or medium quality is rapidly tailing off. So this is an important limitation, I think, to highlight. And if I can go back, so the strengths of this kind of analysing shotgun metagenome data using this kind of MAG approach. I mean, obviously it gives you genomes, and that's unequivocally good. You can predict genes from those genomes, understand what the function of those genes are, so you're getting direct insight into the functional capacity of the species in play, and because you're using Illumina sequencing, the error rate's quite low. I've talked about the limitations of quality, which is basically the next 4 points, but I would also like to emphasise this one, that this is also very dependent, the outcome of this exercise is very, very dependent on the complexity of the microbiome you're working with. A little more challenging to use this data for routine statistical analysis. You have to spend a bit more time thinking about that. And there are some really fundamental difficulties in the field, and one of which is the short-read sequencing technologies limit the number of contigs. They can limit the ability to capture a complete closed genome sequence. And in general, strain-level recovery is still a very, very challenging problem. And most of these contigs are inherently multi-haplotype in genetic terms. And identifying horizontal gene transfer is also difficult using this approach. But what's changing this, and this is really something that's emerged in the last 5 years, is the development of long-read sequencing. So we can see this is a really beautiful diagram, a little bit out of date, but I've annotated some of it. This is basically the read length you get from given DNA sequencing technologies. So Sanger sequencing is here, Illumina sequencing is here. You get like 100 base pairs, 150, 300 base pairs at maximum. And this is the total amount of sequence that's generated from these instruments. So the really large scale, production level Illumina instruments are now off the top of this scale. But they generate a huge amount of this short-read data. But there's a new class of what we call single molecule sequencing technologies typified by Oxford Nanopore Technologies and Pacific Biosciences that can generate about the same amount of data, but the read lengths are much, much longer. They're actually sequencing individual molecules that might be, fragments of DNA that are 10,000 or 50,000, 100,000 base pairs, and you can get them in one go. And this makes a massive difference in your ability to reconstruct genomes from microbiome data. These are early papers of ours in this space from 2018, 2019. These are communities that are bioreactor communities, are a little bit less complicated than the human gut microbiome. But just with even a gigabase of this long-read sequence, we could just get complete closed genomes that just dropped out of the assembly. We didn't have to do any more genome binning analysis than that, which was a great step forward for us. And interesting later studies, we've also been able to show that we can capture non-chromosomal replicons using this kind of approach at the assembly level. Phages, plasmids, and even evidence for multipartite genomes in some bacterial species. So I think you're going to see a lot of movement on the use of long-read sequencing in the next 12 months. I think there's some really interesting studies that have come out this year, and these new sequencing technologies are going to be really widely used. So what about function or direct measurement of function? So this is a really early study from Shunichi Ishii, who was then at UCSD, I think he was at then. And then, so what they're doing here is extracting DNA, if you have a given sample, so extracting DNA, but they're also extracting RNA and sequencing the RNA as well as the DNA from the same sample. And what that lets you do is get measurements of gene expression in genes that are in species in the microbiome you've studied in that sample. And this gives you a very, very powerful view to distinguish between active and inactive states of a given microbe. You know what genes are getting expressed in individual species. So in this heat map, these are different classes of genes here. These are different genomes across the top in different samples. And so you can see these ones here, for example, are expressed, these genes here are expressed in all members of this community. But these genes down here, or pathways I should say, are specific to individual genomes. So there's only one species that's actually expressing these pathways. And then down here we have a more complicated case where it looks like a pathway is actually, its expression is kind of split across two different species, suggesting that there's some kind of interaction going on between these species in relation to that broader biological function. You can extend this idea of co-expression, you can extend this idea of co-extraction to try and extract metabolites as well as RNA and DNA and potentially protein from a given sample. And for example, Paul Wilms's group in Luxembourg has done a lot of work on robotic systems to kind of do this en masse. This is some of our sort of current data on this. We're doing these time series experiments in these communities. We can extract metabolome data, metagenomics and metatranscriptomics, and then we can look at these metabolite profiles, try and map these to compounds, so we know what this compound is, and then in some cases we know what the enzyme is that will use that compound as a substrate or a product, and we can try and examine which species are expressing that gene, and then try and put together a kind of map of these relationships between these different views of the functional state of the community. This is quite a challenging game, and I think again there's a lot of interesting work going in this space. But one thing I would caution is that this idea of integrating all these levels together, of data together, still remains very, very challenging. Particularly trying to understand the relationship between genetically encoded measurements, so either looking at DNA or RNA or protein, if you're measuring metabolites, of course, you're decoupled from that set of natural relationships. And so you're either relying on known biochemistry to tell you what pairings to look for, or you have to rely on sort of large-scale statistical associations to try and uncover relationships between novel metabolites and particular species. But again, I think this will be a very active space in the next few years. So if I can summarise this, I have, I've got a minute left. So I think with studying microbiomes using these culture-free techniques, I think there's often a bit of a false dichotomy between 16S and metagenomics. There's a lot of arguments about whether 16S it could be considered a metagenome technique. I don't think that's really important. I think 16S profiling still remains a very, very useful technique, particularly if you're just getting into microbiome work for the first time. Its influence is waning relative to shotgun metagenomics, and also the cost differences these days are not really that much. Sequencing is very, very cheap. It's the library preparation cost per sample that's the limiting factor in all these studies. And also with 16S, long-read sequencing is making a massive difference, because you're no longer restricted to these short regions of 16S. You can capture the whole 16S gene, or indeed the whole ribosomal operon, and this is a big step forward. For shotgun metagenome data, also I think there's often in the literature this kind of view that is read level analysis or mag level analysis, genome level analysis, the most, which one should be used. Actually, I think for many of us we use both in the same study, because it's there are so many biases there's many biases in the bench side and the extraction process, but also the analysis side has many biases as well, and so you want to get a global view of all this. I think long-read metagenomics is going to make a really, really big impact in microbiome work, but it's probably going to be complementary to short-read sequencing for the foreseeable future. But I think you're going to see very, very rapid progress in this over the next 12 months. And then finally, multi-omic studies where you're measuring these different types of biomolecules in a set of samples. I think this really remains highly complicated, and there's a lot of people out there saying this stuff's kind of going to be routine and so on. But I think technically it's very, very challenging. I think you have to put a lot of effort into experimental design and execution, getting that right. And I think ultimately you have to scaffold all that data back onto a metagenome assembly from your own sample, not rely on reference samples. It's great to be presenting again to a neurogastroenterology community, and I sort of been thinking through how to connect microbiome work with neurogastro. And for example, I think there's a lot of interesting data around now on defecation frequency and stool consistency and so on. And Phil and I were talking about this a bit last night. This is one such study. I'd like to thank my group at CELSI. And finally, just another plug. This is a special issue of the Royal Society Journal of Molecular Omics that's with myself and several colleagues, and it's a special issue on multi-omics for microbiome analysis. And please, it's a great journal. It's a not-for-profit journal, so I think those kind of journals are a great initiative. So please consider submitting a study, and I'll stop there. Thank you very much.
So now for me, this is the exciting part of the session. I still remember as a fourth-year medical student giving my first presentation at a national meeting, and I'm delighted that one of our medical students here at UNSW is doing exactly the same. So Eunice is a fourth-year medical student. She has just concluded her honors degree with us at the Microbiome Research Center at St. George, and her expertise is in the microbiome and dementia. And it's fair to say that over the last year, Eunice has acquired so much knowledge about this topic that if I want to ask anything about dementia and the microbiome, I would actually ask her. So I'm delighted that she's giving the talk today. And her title is Are Bugs Eating Our Memories? The Microbiome in Dementia.
Thank you very much, Professor Alema, for that very kind introduction. And thank you to ANGMA and the organising committee for having me at the conference today. I feel so privileged and I'm so grateful for the opportunity to present. Today, I'd like to give you a brief introduction to the microbiome in dementia, a field that I'm very passionate about and completed my honours project on this year. So I'll start with a very brief introduction to dementia and the microbiome, and then attempt to briefly answer 3 key questions that I had when I first started learning about the field. Who's there? How does it work? And where to from here? Dementia is a chronic neurodegenerative syndrome that results in progressive cognitive decline beyond that of normal biological aging, which interferes with daily life. It can affect numerous cognitive domains, including attention, memory, learning, language, personality, and behaviour. Worldwide, dementia affects 55 million people, with 10 million new cases diagnosed annually. In 2019, dementia was the 7th leading cause of death globally, accounting for 28.3 million disability-adjusted life years, or DALYs, and its prevalence is expected to rise to 139 million people affected in 2050. In Australia, over 400,000 individuals have dementia, and prevalence is expected to double by 2058. Approximately 2/3 of these people live in the community, but of all the residents in residential aged care facilities, 68.1% of these people have moderate to severe dementia. It is also the leading cause of death in individuals aged over 85 years old, and the second leading cause of burden of disease, contributing 242,000 DALYs annually. However, the burden of dementia extends well beyond the patient, with 1.5 million Australians caring for someone with dementia. It also burdens the Australian healthcare system. People with dementia average 20 GP and 5 specialist consultations annually and account for 12% of all MBS services. Dementia also accounts for 25,500 hospitalisations annually, and the length of stay is usually 5 times longer than the average. There are many different causes of dementia. The most common etiology is Alzheimer's disease, which accounts for about 70% of all dementia cases, followed by vascular dementia, mixed pathologies, frontotemporal dementia, and dementia with Lewy bodies. The pathogenesis of dementia remains unknown. However, several hypotheses have been proposed, such as the amyloid cascade, tau protein hypothesis, and genetic contributions for Alzheimer's disease, and the vessel occlusion and multiple infarct models for vascular dementia. Another theory is chronic neuroinflammation, driven by astrocytes and microglia being a trigger for neuronal damage. Sadly, there is currently no cure for dementia. Instead, interventions focus on symptom management and improving quality of life. The limited progress made on these current hypotheses alone has led to the exploration of other possibilities, one of which is the microbiome in dementia and neuroinflammation. As you've all heard, the human microbiome is the genetic material of all the microorganisms found on or inside our bodies. And the microbiome and its interactions with the host phenotype is known as the microbial signature. Dysbiosis of the microbiome, or a disruption to the normal composition of the microbiome, is associated with not only gastrointestinal diseases, but also neuroinflammatory conditions, including multiple sclerosis, Parkinson's disease, anxiety, depression, and dementia, among many others. It's well established that the composition of the microbiome changes throughout life. The microbiome is generally pretty stable throughout adulthood, but in aging, microbial communities become more volatile and diverse. Eventually, however, this stops and results in an overall decreased diversity and less inter-individual variation. In particular, the genera which are normally dominant in healthy adults, like Bacteroides and Lactobacillus, are decreased in older people, and there is instead a proliferation of opportunistic bacteria, like Clostridium. So who's there? Over the last 5 to 10 years, there's been lots of emerging evidence suggesting that the microbiome of older people who have dementia is dysbiotic and different to older people who are cognitively well. In 2017, a study by Catano et al. In Neurobiology of Aging reported that they were the first to identify gut microbiome alterations in Alzheimer's disease. They recruited patients from a larger study involving 18 memory clinics across Italy and included 150 patients with Alzheimer's disease and 150 cognitively well controls. These participants provided stool and blood samples, and they also underwent PET scans for amyloid status. In this study, they identified that cognitive impairment was associated with lower abundance of E. rectali, which is typically anti-inflammatory, and an enrichment of the pro-inflammatory Escherichia and Shigella. Also, amyloid-positive individuals showed higher levels of pro-inflammatory cytokines like interleukin-6 and interleukin-1 beta in the blood. This study was quickly followed up by a similar American study in Nature Scientific Reports, looking at the gut microbiome in Alzheimer's disease. This study found that there was decreased richness and diversity of the gut microbiome in participants with dementia, which mirrored that of other conditions with known gut microbiome changes, like obesity, inflammatory bowel disease, and diabetes. They also identified a distinct microbial signature associated with cognitive impairment, which was characterized by dysbiosis, which likely played a role in disease progression and maintenance through immune activation and systemic inflammation. These changes included a few key bacteria, including a reduction in firmicutes and an increase in Bacteroides, which has also been reported in the microbiome of people with type 2 diabetes and obesity. There was also a decrease in the genus Bifidobacterium in dementia, and Bifidobacterium is normally associated with anti-inflammatory properties and decreased intestinal permeability, with the ability to decrease LPS or lipopolysaccharide levels in the gut. There have since been many studies conducted internationally looking at the gut microbiomes of people with and without dementia, and these have shown 2 key points. Firstly, despite some key taxa being consistently differential for cognitive status, the microbial signature for dementia seems to differ between countries or regions. And secondly, different types and stages of dementia seem to be associated with slightly different microbial signatures. So first, there is only partial consensus of microbial signatures for dementia across different regions and countries. For example, in the US study that I mentioned earlier, there was reduced relative abundance of firmicutes and Bifidobacterium and higher levels of Bacteroides in Alzheimer's disease patients. By contrast, a Chinese cohort of patients with Alzheimer's disease had lower levels of Bacteroides, along with Lachnospira and Ruminoclostridium, and an increased abundance of Prevotella, and there was no difference in richness, but increased microbial diversity. Then a Kazakhstani study found an increased relative abundance of Acidobacteriota, Verrucomicrobiota, Planktomycetota, and Synergistota among Alzheimer's disease patients, and no difference in richness or diversity between the dementia and control groups. And there are many further examples of differing signatures between or even within the same country or region. Although these differences between countries have not yet been fully explored, we know that the microbiome is affected by many different factors, including geography, diet, and culture. So there is the potential for these factors to be influencing the dementia microbial signature, making each country's signature very unique. Secondly, while the large majority of research in this field so far has focused on Alzheimer's disease, recent studies have found that different types of dementia are associated with different microbial signatures. For example, a recent paper from Japan compared the microbial signature of Alzheimer's disease to Parkinson's disease and dementia with Lewy bodies. They found that the dementia with Lewy bodies patients had characteristically higher levels of Ruminococcus torcus and Collencella and a depletion of Bifidobacterium. However, these were unchanged in Parkinson's disease. In Parkinson's, there was a significant reduction in the relative abundance of taxa, which produced short-chain fatty acids. There is also recent evidence suggesting that changes in microbial composition are present even at the preclinical stage of Alzheimer's disease, which is where CSF and brain PET scans already show pathology, but function is pretty normal and symptoms are extremely subtle. A US study published earlier this year identified several taxa which were significantly associated with preclinical Alzheimer's disease. And these included Barnesiella, Odoribacter, Bacteroides species, and Methanobrevibacter smithii. So this suggests that microbiome markers in stool could complement early screening measures for preclinical dementia. It also strongly supports emerging hypotheses about the role of the gut microbiome in dementia progression and potential opportunities for interventions to target the preclinical stage of dementia. So knowing these changes in microbial taxa is an important first step, but it begs the question, how is the gut linked to the brain and how does it all work? The microbiome has this bidirectional communication network between the central nervous system and the enteric nervous system called the gut-brain axis. And this involves multiple connections, including the vagus nerve, immune system, and bacteria and their metabolites. In older age, the combination of immunosenescence, gut remodeling leading to poor barrier function, increased blood-brain barrier permeability, and microbial dysbiosis causes the gut-brain axis pathways to become dysregulated. This ultimately results in neuroinflammation, which is thought to trigger further microglial activation, reactive oxygen species production, and mitochondrial dysfunction, all of which ultimately contributes to cognitive impairment. Although there is limited knowledge on the precise mechanisms underlying this, studies of the gut microbiome in dementia have identified many bacterial taxa, metabolites, and functional pathways which are thought to contribute to this, and I'll briefly give you two examples. The first example is Collencella, which is commonly reported to be enriched in Alzheimer's disease and dementia with Lewy bodies. It is thought to modulate the gut-brain axis at the level of the gut epithelium and contribute to that pro-inflammatory state that we see in dementia. It has also been associated with other conditions, including rheumatoid arthritis, non-alcoholic steatohepatitis, obesity, and atherosclerosis. In a mouse model of rheumatoid arthritis, Collencella was found to decrease the zonulin 1 or ZO1 tight junction protein and thus increase gut permeability. This was then hypothesized to facilitate the exposure of the intestinal neural plexus to harmful inflammatory bacterial metabolites like lipopolysaccharide or LPS and contribute to that neuroinflammation and oxidative stress. Collencella was also found to increase production of interleukin-17A, a pro-inflammatory cytokine in intestinal epithelial cell lines. So speaking of lipopolysaccharides, LPS is another commonly implicated metabolite, which is a component of gram-negative bacterial cell walls. LPS is produced by a number of bacteria which have been found to be enriched in dementia, including Veillonella species. And it works primarily at the level of the blood-brain barrier by increasing its permeability through a number of mechanisms. These mechanisms include upregulating the expression of matrix metalloproteinases, which then degrades the extracellular matrix and digests the basement membrane, destroying tight and adherens junctions, inhibiting endothelial nitric oxide synthase, triggering cell apoptosis, and inhibiting the production of ATP. Another emerging mechanism by which the microbiome can interact with the central nervous system is via the oral microbiome. In particular, there has been increasing interest in the association between periodontitis and dementia. In 2020, a large longitudinal US study examining the associations between periodontal disease and Alzheimer's disease was published. They used data from the National Health and Nutrition Examination Surveys and linked National Death Index and Medicare data with 26 years of follow-up. The authors created these multivariable adjusted hazard models and identified 19 oral bacteria which were associated with increased risk of and mortality from Alzheimer's disease. And these included very common periodontal pathogens like Prevotella and Porphyromonas species. Since then, several more studies have highlighted a similar association between periodontitis and dementia. Given the likely association between peripheral inflammation and dementia, some studies have even suggested a mechanistic relationship between the two diseases. Interestingly, there was a study from 2013 which looked at post-mortem immunofluorescence labeling of brain tissue from 10 people with Alzheimer's and 10 controls. And what they found was that in the brain tissue of 4 of the 10 Alzheimer's disease patients, they consistently were positive for LPS from P. gingivalis, which is a key periodontal pathogen, while all the controls were consistently negative. So this was compelling evidence to suggest that periodontal bacterial metabolites can actually access the brain during life. And this translocation of inflammatory metabolites could induce or exacerbate neuroinflammation and contribute to dementia pathogenesis. However, very few studies on a similar topic exist, so it would be very interesting to see whether these results can be replicated in the future. This hypothesis about periodontitis contributing to dementia is also important because we know that individuals with dementia often have poorer oral health and higher rates of diseases like periodontitis and gingival bleeding. And this can be due to a combination of different factors, including a deterioration in executive functioning and motor functioning, a poorer diet, and poor oral hygiene education. So there is potentially a bidirectional relationship between dementia and periodontal disease mediated by the oral microbiome. Since periodontal disease is largely treatable, further investigating this relationship could reveal a potentially modifiable risk factor for dementia. So where to from here? Despite all the research that already exists, this field is still very new and there's still a lot to explore and learn about. One key gap in the literature is that so far there have been no comprehensive studies investigating the microbiome of older adults with dementia compared to those who are cognitively well. And this is an incredibly important topic to explore because we know that factors like geography and environment can significantly impact the microbiome. So that means that although we might know the dementia microbial signature for many other countries, the Australian one could be very different. This is actually an area that the MRC's Healthy Optimal Australian Microbiome Study is currently investigating, and I had the privilege of conducting a pilot study on this with some very promising results for my honours project this year. So it'll be very exciting to see what the full results from the larger study will show. This field is also moving towards a multi-omics approach to analysis. So while it's great that we know which bacteria might be associated with dementia, it's important to further our understanding of the potential functional pathways and metabolites involved using technologies like metatranscriptomics, metabolomics, and proteomics. Of course, the ultimate dream is that better understanding the microbial signatures of dementia will enable the development of personalised microbiome-based dementia prevention and therapeutic strategies. While there's a lot of work that is ongoing in this area, mouse model studies of longevity and cognition have already taught us many things. For example, a 2021 mouse model confirmed the idea that a youthful microbiome contributes to longevity. In this study, the authors performed fecal microbiota transplantation, or FMT, from young mice to older mice, and showed that the older mice, which received the FMT, had significantly improved cognitive function, learning abilities, and immunity. Another very exciting study that was published just last month showed that the microbiota from patients with Alzheimer's disease induced deficits in cognition and hippocampal neurogenesis when transplanted into microbiome-depleted young adult rats. Importantly, the severity of these cognitive impairments in the rats correlated with the donor patient's clinical cognitive testing scores. And that was the first time that Alzheimer's symptoms have been shown to be transferred to healthy young organisms through FMT. And it importantly confirms a causal role of the gut microbiome in Alzheimer's disease. So in summary, there's a lot of evidence suggesting that there is dysbiosis of the microbiome associated with dementia, and that the composition of the microbiome appears to vary by type and severity of dementia. Regardless, the microbial signature of dementia is commonly dominated by pro-inflammatory bacteria, which may modulate the gut-brain axis at various levels through the metabolites they produce to induce neuroinflammation and contribute to dementia progression. Bacteria, which is commonly implicated in periodontitis, are also commonly enriched in dementia, revealing a potential role for oral disease and perhaps even other conditions to contribute to neurodegeneration. Ultimately, it is critical that we better understand the microbial signatures in dementia, as this will pave the way for the development of personalised microbiome-based dementia prevention and therapeutic strategies. So I will leave you with a little bit of food for thought. Perhaps bugs are the ones eating our memories, but they could also be the key to solving dementia. Thank you very much.
Thanks much, Eunice, and thank you all the speakers. Wonderful presentations. I hope you've been, enjoyed the presentations, and we look forward to having questions from the audience, please. So feel free to.
Hi, Lincoln Stan from the University of Melbourne. Amazing session. Thanks so much for this. It's gone. A bit of a naive question, I guess, for Georgina and Rowan predominantly. Are people trying to make, trying to produce bugs? So basically, producing bugs that are specialised in a dish before you transplant them. So doing like an in vitro version of phage therapy.
Yeah, absolutely. So as I was alluding to, it's about understanding what's needed in the specific situation and understanding what an individual needs in that specific situation. So yeah, I anticipate that ultimately it will be a unique combination for an individual with unique properties that are needed. So, and that is what's starting to happen. I can hear you and I can repeat your question.
My question is for Eunice and also Professor Ho. You mentioned about oral microbes, and I think it's fascinating because it's associated with Alzheimer's disease, but also cardiovascular disease, autoimmune disease, several cancers. Do you think that it actually, this is the part where it has a lot of hidden germs, where you'll find association and clues to many different diseases? And biologically, do you know whether there are established mechanisms how the oral cavity is involved with cognitive function?
Absolutely. It's a no-brainer not to look at what's going on in the oral compartment. And a lot of the studies that we've set up at the Microbiome Centre have had that in mind right from the beginning. So my IBD study has always been recruiting, collecting samples from the oral microbiome as well as the gut, because the microbes in your gut have to get there somehow, and the majority of them are going to get there from the journey at the top of the intestinal tract. So, and we know that the more we look into whatever conditions, whether it be intestinal problems, IBD, colon cancer, all the information that's coming out about dementia, and the oral microbes are coming up in those signatures of risk. And so understanding whether they are doing different things because they are in a different environment when they're down in the intestine compared to in the mouth is really, really fascinating. Some of the work that we've published has tried to understand that, where we've isolated the same organisms from the mouth and from the gut and tried to see if they have altered functional capacity just because they're in the wrong place at the wrong time. But it's absolutely important.
Yeah, I 100% agree with what Professor Holt has said. And in terms of the literature on dementia and Alzheimer's disease, a very similar thing has been found, where pathogenic bacteria from the oral cavity is consistently coming up when people look at the gut microbiome signatures as well. So many people have assumed that it comes from the oral cavity and is then just being picked up in the stool samples. In terms of like a mechanism, I don't think it's been like fully confirmed in dementia either, although in the study that I was referring to where they found like the LPS and other bacterial metabolites in the brains, like post-mortem, they were found like along the trigeminal nerve, so they presume that it might be somehow related to like traveling along the nerve, although we don't exactly know at the moment.
I think the location is quite key. I mean, the fact that it's physically closer to the organ of interest, and the fact that there is inflammation, there's chronic inflammation. When you have inflammation and a breach of barriers and stuff, it's an open game. So I think it's underrated. The oral microbiome is actually very, very important. And I think if you're doing any microbiome research, please collect oral samples. You'll be surprised how relevant these are.
Can I just add to that? The other caveat is that we're trying at the moment to apply our multi-omics approaches to oral samples. This is globally, with the knowledge that we have from the intestinal tract, and it's different. We know that community-wise it's different. We know structurally, functionally-wise it's different. So at the moment there's a lot of studies that are trying to look at the oral microbiome and they're saying there's no signature there. Well, actually there's not a signature if you apply the constraints of what we try to do in the intestinal tract. So there's a lot more to understand in the oral microbiome. So although there's a lot of negative studies out there, we haven't got the technology right yet. So I encourage people to not dismiss it based on the negative studies coming out at the moment.
Thank you. From Adelaide. I'm just making a comment that the great Maggie Beer in Adelaide has had a campaign on the inadequacy of the diet provided in aged care. And she's a wonderful cook from Ross Valley. And I think that's almost certainly true. And in fact, I think there probably has been an uptick in people in aged care having a choice between not getting the yellow mush and the orange mush is there, which one do they have to feed? But the other thing that people tell me is people's oral health goes down the tube very quickly in aged care. And it struck me that if maybe you could do a intervention study by trying to get the diet good and make sure everyone gets their teeth cleaned once a day. And it'd be interesting to see what impact that would have. And it's one sort of ethical intervention you could do and then monitor outcomes. It's just a thought.
Thank you. Did people manage to hear that at the back? Yeah? Okay. I agree. I think... do you want to take this?
Actually, I can answer this because, I strongly believe in exactly what you say. I think, the nutrition in old age, especially in institutions, is really, really awful. And that's when you want your diet to be optimal and your microbiome to be optimal. There has been such a clinical trial, not necessarily in a setting. This was in the context of frailty, and it was actually adopting a Mediterranean diet for a year across 5 European countries, and showed that in fact, if you eat a healthy diet like a Mediterranean diet over a prolonged period, it does impact very positively on all markers of frailty, but also improve your microbiome. So there is already some template for that, but the context that you outline in old care, facilities, that would be a very easy study to do, and it's very, very worthwhile, and incorporating something like the oral hygiene would be very important as well.
The other thing, just to add to that, is the impact on the oral microbiome by the changes that occur due to the loss of teeth and the implementation of dentures and stuff like that, which I know is not specific to the aged care facility, but it's obviously something that is going on more and more in that age group. So that's also really important for us to understand. Plus the increase in orthodontics and the use of that more and more younger people are having as well. So this is all part of the process.
Thank you.
I'd like to raise a question about gut transit. We're in a motility meeting and we have to bring transit into it. And it has relevance for the ageing population and the Parkinson's population. And it leads me to think, traditionally, and in Darwinian survival terms, you get infected with a parasite or a gut pathogen, you get diarrhea. And one of the thoughts is, well, you have more rapid clearance. And So I'm just trying to, in a complicated way, think, a fundamental question here, do we know whether the constipation in Parkinson's or in the geriatric population may be impacting the microbiome signature or the dwell time or gut permeability or transgression of the membrane by the bugs? And it would be a simple study to do to look at the impact of transit time and manipulating that on these a number of these variables. Just sort of comments and questions.
No, it's a very good point.
Okay.
No, Ian, it's a very good point. I think I would cringe to define what that clinical study should look like.
But that study's been done. I mean, in fact, the paper publishing got just a few weeks ago, precisely on Parkinson's, and showing that, in fact, it was in the news all over the place because it was suggesting that, in fact, Parkinson's may actually start in the gut, going back to Hippocrates and what we said yesterday. And you're absolutely right, Ian. I think it's the gut transit, whether this is actually an inherent problem with Parkinson's that precedes the presentation of the classic symptoms and how it's linked with the microbiology of the gut. This is now an open game. I think it's going to be very exciting because people will address precisely your questions and try and figure out, cause and effect. But there's very, very promising data that, there is an origin of Parkinson's in the gut. So I think that's very exciting that's coming through. Sure. I was just waiting for how long it would take Ian Cook to talk about the hyperparameters. Yeah, so it didn't take long. Thanks. Absolutely, Ian. I agree. 100% agreement. Yeah. I have a question. Sorry, question from the audience again. Yeah.
Yeah. Thanks a lot. Look, it's about this discussion. Is there anything known about the rates of dementia with regard to countries that have good or bad oral health? Might be good to look at something like that. The other thing, just to add another layer of complexity, just to get the conversation going, is my understanding, not working on the microbiome, is that it's never stable during the day. It's changing between day and night. So a lot of what we're talking about, I don't see many caveats about when samples were taken. And do you think that is important?
I think it's easier to start with the second point, which is you're absolutely right. It's not a static entity, and so consideration for how and when and what to sample is really, really important. A lot of the studies, why we do longitudinal studies, is to allow us to see that natural variation within a health and then be able to map that into a disease context. So we know that you need to have probably 6 or 7 repeated samples across duration to actually get to a sort of standard understanding of what is normal variation. But when you compare that in healthy individuals to disease cohorts, then that's when we're looking and seeing the most informative information about that. But understanding and factoring in things such as circadian rhythms, antibiotics. We know that the microbiome will change within 24 hours of a dramatic nutritional shift. So yes, all of these things have to be factored into the studies, but even when you do all of that, there's still really important microbial factors that are coming through. So you have to be cognizant of that. And that's why some of the studies that we see some very unusual results are probably due to poor study design or small numbers or limited longitudinal signalling. And that's fundamental to and why you need to collaborate with people who know what they're doing with the microbiome to get the best out of your clinical studies.
But it's important to add that those minor changes that happen during the day, diurnal or diet-related, are really insignificant compared to the difference with the disease signature. So all of us, if you eat a Big Mac this morning, your microbiome is going to change, but it's not one that if your fundamental diet is healthy, you will probably maintain that and you have little blips But if you compare that to somebody with inflammatory bowel disease or some other condition, it'll be dramatically different. So I think those minor changes are not, I mean, we have to be cognizant of them, of course, and account for them, but it's not a reason for not trusting the difference between that and a disease signature, because that's really much more dramatic.
And there's also, there's many physiological systems that are variable. I mean, the immune system has massive variation in the types of cells that are getting produced in a given time interval. There's also, I think, some interesting analyses that are starting to emerge, a much more nuanced interpretation of what taxonomic profile data actually means in terms of trying to infer growth rates and other kind of responses that are more physiologically informed. There's a nice paper by Sean Gibbons from the Institute of Systems Biology in Seattle in Nature Comms, I think a couple of weeks ago, that sort of starts to dissect this from data. So I think there's a lot more subtle interpretations that are starting to emerge. But also Ian's point, I mean, I think there's still so much that's not known about what's actually happening in situ in the lumen, because it's so difficult to measure. And I mean, I think I was having a discussion recently with someone about the microbiome in NASH and the influence of the microbiome in NASH, and some of the ideas that it may not be like really specific signals of particular bacteria. It could just be, is there an enrichment for more gram negatives, and then so you're presenting more LPS, for example, and the immune system's seeing that. And so I think there's probably like an interesting, relatively coarse level of analysis that would use this kind of functional data that could be interpreted in more creative ways that might be an interesting direction to take, along with better measurement techniques in situ.
And the last bit of your question was about dementia rates in oral health, high and low quality. Do you have anything?
I think a lot of my points have already been covered, and I'm not personally aware of any data on this, although there may be something out there that I just haven't seen yet. But I think, again, because the oral microbiome is so variable, like we've mentioned, it could, after you brush your teeth, it could be different, or after you change your diet, for example, or if you go into residential aged care facilities, your diet and nutrition might change completely. So I think it would be interesting to see, how we can standardise that, and how we can measure what a quote-unquote standard oral microbiome would look like between the different countries. But I think that's a really interesting point. Thank you.
And I may just add, there's a study that hasn't been published, the privilege of being an editor, sorry, that looked precisely at this, of how can you change your gut microbiome by changing your oral hygiene. So if you take a mouthwash, for example, it does affect what appears in your gut analysis. So to some extent, this is a brand new area, I think, that deserves a lot more attention and probably a lot more collaboration with dentists. They don't feature in any of our studies, yet this is an important kind of organ that should be certainly incorporated in our studies. So Howard, do you want to conclude?
Yes. Sure. So in the interest of time, I think it's better to conclude now. So I would like to once again to thank the three speakers for their wonderful presentation that sparked a very interesting discussion in the floor. So I think it's time for us to feed our microbiome in our morning tea. So please join me to thank all the speakers. And please come back at 11.